Hi again, everyone. Welcome to our discussion on Chapter 4, which is about carbon. In some ways, this is a preview of what you might encounter as you begin organic chemistry. It will also dovetail with some themes that you would explore in biochemistry, both of which strike terror in the hearts of students sometimes, but shouldn't. It's something that's fundamentally part of our lives, and we don't even realize it sometimes, just like water. So this chapter, we're going to talk about the properties of carbon and some of the things that carbon can form. We're going to be doing a lot of terminology in this section. So as always, if you have any questions, reach out to me, and I'll be more than happy to try to help you find answers. So the study of carbon in its chemical properties is fundamentally organic chemistry. It doesn't have to be, it doesn't have to come from a living thing. Oftentimes it does because living things use carbon so much of the time. But organic chemistry is the study of compounds that contain carbon. So we can think certainly of the biomolecules that we have, nucleic acids, proteins, carbohydrates, and fats. The salts that we've talked about in earlier lectures, along with water and oxygen, even though they are incredibly important and necessary for life, are not fundamentally organic molecules because they do not contain carbon. Now, if you say vitamins, now vitamins are often organic compounds that serve as cofactors for proteins, and we'll get to that in another lecture. But in any case, we have organic things which contain carbon and inorganic, and they don't have to come from a living thing. You can make these things artificially. They can be as simple as a carbon with 4 hydrogens covalently bonded to it, or like a caffeine molecule, for instance. This has got carbon and nitrogen. Where you see an angle bend right there, there's a carbon that's inferred, like right here, or right here, or here, or here. So you can put a letter there, or you can be at a place like this. One thing you'll notice where you have an unwritten letter, you'll see 1, 2, 3, 4 lines emanating from that letter. And that's because carbon has a valence of 4. Just like it has 4 covalent bonds with hydrogen, this carbon right here has 2 covalent bonds to oxygen, 1 to this nitrogen over here, and 1 to another carbon over here. It can be a little more complex like that, or it can be almost infinitely complex. All these little angles here and there are carbon atoms. These O's are oxygens, obviously. Some nitrogens down here. The sky's the limit with carbon. It can form long, long polymers. Virtually unlimited structures can be created from carbon. But we're not going to go there today. We're just going to basically try to get you the basics. Most organic compounds contain hydrogen, like this, and you can see hydrogens here in addition to carbon. That's very common. This is a glucose molecule, but oxygen and nitrogen and phosphorus are all somewhat common as well. But the 3 biggies would be carbon, hydrogen, and then oxygen. And then you'd see nitrogen and other ones. So things like a glucose molecule, these are all carbons again at these angles here, or something like what we call a fatty acid, whether it's saturated like this case is, or it has no double bonds, or unsaturated where it has, in this case it has 1, or you can have 1 or more double bonds. So lots and lots of possibilities with organic compounds. What makes carbon so special and relatively unique? There are other atoms that have carbon's valence, what we call a valence of 4, meaning that it has 4 reactable electrons, normally reactable electrons, so you can see the 4 dots around this. Silicon is another element that has that valence of 4, but we have yet to find a true silicon-based life form. There may be some aspects of electronegativity or something that we haven't accounted for yet, but carbon and silicon are really pretty close to being the only atoms that are really hypothesized to be necessary for life as we know it because of that 4 valence. And that's what we're getting at here, this electron configuration. That's the structure and function relationship with an atom. determines what kinds of bonds will form with other ones. With a valence of 4, we can do lots of things. Turns out nitrogen's in there quite a bit as well, oxygen, so forth. These guys over here form functional groups that react with the carbon. Oftentimes we have a basic skeleton, a structural skeleton made up of carbon, and then nitrogen, oxygen, hydrogen, and these other elements that we've talked about fill in terms of reactive groups. They add on to the properties of that molecule. So here's our carbon. Again, it has a valence of 4. It can form up to 4 double or covalent bonds, maybe, or a combination. So you could have 2 double bonds, or you could have a triple bond and a single bond. That's all theoretically possible. And it's not planar like this. When you look at it, it looks like a 2-dimensional object in this case. It's actually a 3-dimensional object. And so one of the things that you learn in organic chemistry, particularly early on, is how to understand the 3-dimensional nature of organic compounds, because they're not just flat like this. They're actually tetrahedral and then branch off of each other in 3 dimensions. And this is more of the same, just showing you that the total number of covalent bonds around a carbon is almost always 4, unless you have some very exotic molecules. In biochemistry, you never see any more than 4. 4 total bonds. That's to say either 4 single bonds, or 2 single bonds and a double bond, or maybe a triple bond. It's rare, but it still happens. Acetylene gas, for instance, which you might have heard about, is something that they use from time to time. That has a triple bond in it. And it's just more of the same again. Again, I don't want to beat this to death, but it's showing you the 3-dimensional aspect of these molecules, that this isn't flat. It's actually 3-dimensional. Now this one right here, this reminds me of a good point. You see this single bond right here? See that right here? And we find it right here. That bond acts like a swivel. So these 2 pyramids can actually spin. Okay, if you had imagine if you had a set of Tinker toys. These do not have to all be the same element or atom. They can be different, and they can actually spin relative to each other. But this guy right here, this double bond is very, very rigid. This molecule does not spin in that plane. In fact, this one, is very planar if you look at it, because of that double bond. It doesn't have that spinability, if you will. That double bond is somewhat rigid, and that plays a very, very important role in the structure of some molecules that we're going to talk about. One thing this slide doesn't mention that I should probably put in here is that the biomolecular families, that is to say carbohydrates and proteins and nucleic acids like DNA and RNA, are polymers. They are polymers made up of carbon chains. That is to say, the backbones of these structures are carbon. Now, fats don't form polymers as much, but they do form these carbon chains just the same. So when we have fats, what we have are a big collection of these types of what we call triglyceride molecules that may or may not be bonded to each other. But these are long carbon chains that vary in length and can vary in shape, and that's true across all 4 groups. It's just that the fats tend to be like this, whereas you can create virtually endless chains in things like peptides or nucleic acids. One group that's very, very common in biological systems, and is why when we drill for oil, what we're doing is we're drilling for the remains of organisms that lived before us, whether they were plants, primarily plants, or animals, because that oil and coal under the ground has deteriorated to the point where it forms lots of very simple organic molecules that are almost entirely carbon and hydrogen. Hydrocarbons, so a carbon backbone surrounded by hydrogens, and sometimes there are double bonds in here as well. So they're consisted of only carbon and hydrogen. Those chains in the fatty acids that I showed you, the triglyceride has hydrocarbon chains on one side of it, and I'll show you more of that down the line. They do contain lots and lots of energy, and the reason is because they have long chains, lots of bonds in here. There's energy stored up in these bonds. And what happens is when we break down things like lipids, what we're doing is we're actually oxidizing these bonds to break them to create energy for making ATP. So here's our triglyceride again. Again, we can oxidize these bonds sequentially in order to harvest energy out of them. So here's a diagram showing you a lot of the molecules that we would find in something you're probably quite familiar with, and that's gasoline. Gasoline is a mixture of hydrocarbons with a little bit of oxygen from time to time and so forth. But in any case, hydrocarbons, lots and lots of energy. It's a very, very energy-rich mixture in gasoline. Lots of energy released when we oxidize it. So here's a picture of, on the right here, a triglyceride with 3 fatty acid chains on the end. And what happens is those hydrophobic van der Waals forces in these hydrocarbons allow for these things to form large droplets. So these fat droplets here are a bunch of these molecules that are associating via their hydrophobic van der Waals interactions. That's one of the reasons why fats don't necessarily need to polymerize like the other 3 groups, because they'll actually sort of form these aggregates on their own, especially in aqueous situations. One of the little tricky parts in understanding carbon chemistry is having a naming convention for all the different possibilities of carbon structures, even with relatively few carbons. That property that even small numbers of carbons can form different structures, even with the same chemical formula. They can have the same number of atoms, the same number of elements, but they can have different structures. We call those isomers. So they have the same chemical formula, but different shapes and structures. And sometimes they'll have different properties because of those changes in shapes. Not usually those differences are subtle, but nevertheless they are. It is a possibility, because when you change the structure, you can potentially change the function. So a structural isomer is one that has the same number of atoms, the same number of elements. It's just created in a slightly different shape. So here is 1, 2, 3, 4, 5 carbons. That would make it a pentane. Well, this is what we would call an isopentane. It's 2-butane is what they would call that, or 2-methylbutane, but I don't want to go there, okay? I'm thinking of the naming conventions that you learn in organic chemistry. But in any case, all 5 of the carbons are arranged linearly here, but we obviously have a branch on this one. So these guys are structural isomers. This one's a little trickier concept. It's called a cis-trans isomer, and in order for it to be a cis-trans, generally speaking, you have to have a slightly different spatial arrangement, and most of the time we're talking about double bonds. So let me move that out of the way here, and now we have our ethane or ethyl group here, 2 carbons. So we have a carbon, double bond carbon. Now I want you to notice where the X's are. So we have the X's on the same side. Remember, that double bond is very rigid. It doesn't spin. All of these single bonds up here would spin like little helicopters, if you will. So it's a very wiggly, like a freshly boiled spaghetti noodle. Okay? These guys down here, much more rigid, much more planar. Even if you have stuff going off of these things, if X means a whole bunch of other atoms. Nevertheless, this bond is rigid. So X and X, this is what we would call cis. Now, if I turn that sideways, I wish I could, I can't do it right now, you would see the letter C. Okay, that would be the letter C if we turned it the right way. Okay, that's a cis isomer. Over here, you notice the X's are on the opposite sides of the double bonds. This is called trans. You might have heard of all trans fatty acids and that sort of a thing. Plays a role in this. They're talking about the double bonds in the fatty acid chains, and those double bonds, the resulting carbon structure, they're on the opposite sides of that double bond. Whereas a cis fatty acid, the carbon chain would be on the same side of that double bond, cis and trans. Then we have our last term here. So this is a very term-heavy slide. It's called enantiomers, and this is really important from a bioactive and pharmacologic standpoint. Turns out we live in a left-handed universe in terms of life on this planet. Well, I'll get to that a little bit later. But if you look at this carbon right here, I wish this is one of those times I wish we were actually in class together, because this is easier to show in person. This carbon right here and the 4 single bonds it has around it, each molecule that it's bound to is different. Okay. If we look at this one over here, it has the same functional groups attached to this carbon, but it's in a mirror image. Okay, if you tried to place the different groups, what you would see is you wouldn't be able to line them up properly, because these are mirror images of each other. You see that the nitrogen is the amine group is sticking out. Notice, and they don't do a very good job of it here, but they are mirror images of each other. Okay? So just like your hands can't superimpose if they're faced the same way. So put your hands out in front of you, between you and the screen, and then try to create, they either have to face each other in order to overlap identically, but they can't do it if they're faced in the same direction. And if you experimented with these molecules in person, which I typically bring a model, you can't do that. You can't superimpose them if they're facing in the same direction. Okay, that's what we mean by enantiomers. It's an organic chemistry term. But it's an important one because many molecules are bioactive based on whether they're left-handed or right-handed. And I actually mentioned this in our first lecture about the fact that morphine is very, very bioactive, but dextromethorphan, again, that infers the right-handedness of it. Dextromethorphan is much less biologically active. This is really, really important in biochemistry. It's really, really important in pharmacology, because if you're generating a molecule in the lab to be used as a drug, if you have an even mixture of these, you might lose potency because you got a lot of this. And how do you get rid of this when it's identical in terms of its chemistry? So they may have very different effects. One may have no effect at all. That's very true. If you look at dextrose, which is a right-handed type of sugar, as opposed to the levo version of it, the levo is not usable, but the dextrose is. So usually only one isomer in this way, usually only one enantiomer, I should say, is biologically active. Different effects demonstrate are sensitive to even subtle variations, and so it can be just the difference between a small little thing that can make all the difference in terms of its activity. And here's some examples, like for instance, ibuprofen, a really lovely painkiller. The effective one is S-ibuprofen, but the R-ibuprofen is inactive, so the mirror image of it doesn't work. Same thing's true with an asthma drug called albuterol. The R-albuterol is active, the S-albuterol is not. And back to our structure-function relationship. As we modify organic molecules, depending on their carbon skeleton, the length, and the three-dimensional structure, but now let's add a dimension to this. We've talked about length, we've talked about isomers. Now let's start adding, shall we say, Christmas ornaments to our Christmas tree. Okay. Trees look very, very different, right? Depending on how many branches they have and then how many leaves they have. Or Christmas trees can look very, very different depending on what ornaments you hang off of it, right? That's kind of how functional groups work. And so we're getting at functional groups. Just as an example, if we had let's say we had a fetus in the womb and we could somehow manipulate which hormones it's exposed to. If we expose that hormone to a lot of that fetus to a lot of testosterone, what we would see is the development of male characteristics. And we can even see this in adults too. Folks who are undergoing gender reassignment, one of the things they'll do is they'll start taking the hormone that is most associated with that gender, male or female. So if we begin giving a testosterone to a person or something, we would begin to see the development of male-like characteristics. Whereas if we give estradiol, which is estrogen, to somebody, they would assume more female characteristics. In fact, there are some disorders out there where you have too much of an enzyme. This one's called aromatase, and what it'll do is it'll convert testosterone to estradiol. And so people who have excess aromatase may be genetically male, but may have many more female characteristics. But the difference between the two are very, very subtle because they both have that ABCD ring, like cholesterol, ABCD here. But it's the functional groups that really make the difference between Mars and Venus. It's the functional groups. So we have a little methyl group right here that we don't have here. We have an OH right here. But other than that, they're virtually identical. They both have a methyl and an OH up here. Okay? But this one right here has that functional group and that functional group. That's all the difference between Mars and Venus. Pretty amazing stuff. Those functional groups have reactivity and they confer function by virtue of their chemical properties on these organic molecule structures. So functional groups superimposed on the carbon backbone are what are most commonly involved in chemical reactions. So functional groups are atoms. They can be individual atoms or they can be small clusters of atoms forming a small molecule that is bound to that carbon structure that give that molecule added reactivity, if you will. So each functional group has a unique properties. Some of them are very, very polar. Some of them are very, very non-polar. They confer the chemical properties on that carbon backbone. And let me give you an example here. Notice here that all three molecules have an NCC arrangement, NCC. That's really the heart of the backbone right there. On one side, that N has two H's, and so we call that an amine, or sometimes called an amino group. Very distinct chemical properties. On the right side here, the COOH is known as a carboxylic acid, or a carboxyl group. So a carboxylic acid, amino acid. So we have this backbone here, and what we do is we bind between these groups and these groups to form long peptides. But notice that we have different amino acids based on what goes right here. R means variable in organic chemistry, means we could have almost anything there. In our case, there's up to 21 or 24 different things that we can put here. We can put like a methyl group, CH3, or we can put a methanol group like a CH2OH. Gives these two amino acids different chemical properties. This one's a little more polar, a little more aqueous. This one's a little more non-aqueous, a little more hydrophobic. And so functional groups confer reactivity. So functional group, and then functional group. One or more atoms that confer chemical properties when they're associated with a carbon compound. So here are some examples of some very, very common functional groups that we find in biology. And if you intend to take organic chemistry or biochemistry down the line, cell biology, this would be great for you to get to know. The more you know in terms of being able to see these in your mind's eye, the stronger you'll be and the easier it'll be to learn the stuff down the line. A hydroxyl group, OH. Carbonyl, C double bond O, and its close cousin, the carboxyl, COOH. Carboxylic acids, they can ionize and be very polar. Carbonyls are usually not quite as polar, but nevertheless have some interesting properties. Like for instance, but if somebody is an insulin-dependent diabetic and they haven't had enough insulin, What usually happens is they start burning peptides in order to make ATP, and one of the metabolites of that is are what are called ketone bodies, and acetone has a very, very fruity smell to it. So it smells like they've been chewing juicy fruit gum when they're actually suffering from the fact that they can't get glucose into their cells. In any case, we have hydroxyl, carbonyl, and carboxyl, all very common. One more slide for you. Again, the more you can understand these and see these in your mind's eye, the easier material, especially over the next couple of weeks, and then if you go to organic chemistry, will be. This is an amine group, NH2. We'll talk about amino acids. So here's the amino group in an amino acid. Here's a carboxyl group, COOH. Sulfhydryl, SH. We use sulfur in this sulfhydryl groups a lot of the time to help stabilize protein structure. We're going to talk about covalent bonding in protein structure, and one of the covalent bonds is called a sulfhydryl disulfide bond, where two sulfurs bond together, so SH, sulfhydryl. Phosphates, very, very common, very, very important in things like ATP. Very, very polar. And we actually use these to change the shape of proteins in real time. By tacking on phosphates or removing phosphates from proteins, you can fundamentally change the shape and enzymatic activity of a protein. A methyl group, CH3. So we put that on there, very nonpolar, very hydrophobic. So here's an example of a methyl group on this, this ring structure. It's actually a part of a nucleotide. So just a small example. Let's call this a case study in functional groups. ATP is very, very important as a source of energy. It's also a ribonucleotide, but we're a little ahead of ourselves here. ATP, that's the molecule that we use to transfer energy from the nutritional sources of energy to the cellular utilizers of energy, shall we say. It's an intermediate molecule between the energy sources and the places where we use energy. So we transfer energy through ATP, and it turns out this right here and this right here is very, very important for that energy storage. But here's where the energy is stored, out here in these phosphates. Has something to do with electron resonance. That's why ATP is very, very useful for storing energy. There's something about it, and Dr. Brunel has a really interesting talk about why the adenosine in ATP is a wonderful energy storage molecule. But these phosphates here, this alpha phosphate, the first one, this beta phosphate, and the third one, the gamma phosphate. These bonds in between the phosphates contain lots and lots of energy. And there's a lot of energy right here. So when we hydrolyze this, we release a given amount of energy. There's even a little bit less energy in this one, but nevertheless, you can hydrolyze that as well to harvest energy. And so this is one of the most common energy transfer molecules in biology, ATP. And it's all about the functional groups. Functional group out here with, carbon backbone and other functional groups. Carbon backbone here with its functional groups. And then we've got 3 functional groups tacked on to that. And I just got another way of picturing these things. So here is this adenosine right here is a combination of adenine and ribose. So when we say adenosine, what we're saying is this part of the molecule, adenine and ribose. You might hear every once in a while, you'll hear ATP is called by its formal name, adenosine triphosphate. There's more energy here than here, and there's more energy here than here, but you can actually harvest that energy as needed in the cell to keep functions going. In the time that you've been with me, which is just shy of half an hour, you have burned a few pounds of ATP. What I mean to say is you've hydrolyzed that gamma phosphate, usually with using water. We call that hydrolysis. We've removed that third phosphate and released energy in order to fuel reactions. Now, over the course of a day, you'll burn somewhere around 150 pounds of ATP. Well, you're not making a new ATP molecule every time. That's why some of you who may not weigh 150 pounds can still do it. What we do is we recycle. So we make ADP and sometimes AMP, adenosine monophosphate, and then we use processes to put a new phosphate back onto that and store the energy in there. Those ATPs are sent out into the cell, and when they're needed to facilitate a chemical reaction, we'll use the energy stored inside that ATP right in that bond in particular to fuel that reaction. This is an example of what we call coupling. We're coupling energy requiring reactions with energy releasing reactions, but that's not in the class right now. That's for another time. But that's it for today. We're right exactly at half an hour. As always, please send me your questions. Let me know what you're having a tough time with, and I'll be glad to discuss issues with you further to help you get a master on this. It can be very, very simple but deceptively complex down the line. So getting these simple tools down first will make your lives in the future much more easy to master the more complex material. Good luck and thanks for your time, and I will talk to you soon.